Hi. In today's main lecture, we're going to continue with a look at the muscular tissue. The focus today is really going to be on how muscle cells contract and the role of the nervous system in making our skeletal muscles contract to produce movement. So, throughout this, we're going to stop and watch some kind of little video clips that break down the process of muscle cell contraction at different points. You can also always rewatch these videos. They are, if you go into mastering, they are in the study area under the video section, and I'll show you that in a minute too. But as you're watching each of the steps of muscle cell contraction, you want to kind of think about, take notes on the answers to the questions as we kind of break down this process. There's kind of a nice overview of the entire process here in this graphic on the right. But in order for a skeletal muscle to contract, we have to have a nerve. So here you can see the nerve. Now I know we haven't covered the nervous system yet, and it's coming up pretty soon in the course. But nerves send signals to our muscle cells to contract. And so the first part of the process we're going to look at is what happens at what's called the neuromuscular junction. So right here, where a nerve makes a contact with our muscle cell, that is the neuromuscular junction. And down here in this graphic, we have taken that neuromuscular junction and kind of blown it up so you can see a little more detail in what's happening. So as you're watching the video, pay attention to what events happen in the nervous system and what events happen at this junction between where the nerve meets the muscle. So to orientate yourself, everything kind of yellow in this picture, right? That's a nerve. Specifically, it's the end of a nerve called an axon terminal. And then where it's meeting the surface, this is the muscle cell. So let me exit out of here. And we're going to go into the video clip. The video that we're going to be watching first here it's one of the AMP flicks. So it's events at the neuromuscular junction, part 1. So in our study area, whoops, not what I wanted to do. If you click on events at the neuromuscular junction, and we're going to watch this video once, and I would suggest that maybe on your own you watch it a few other times and stop it and take some notes.
Nerve impulses, also known as action potentials, travel from the brain or spinal cord to trigger the contraction of skeletal muscles. An action potential propagates down a motor neuron to a skeletal muscle fiber. The site where a motor neuron excites a skeletal muscle fiber is called the neuromuscular junction. This junction is a chemical synapse consisting of the points of contact between the axon terminals of a motor neuron and the motor end plate of a skeletal muscle fiber. The events at the neuromuscular junction occur in 7 coordinated steps. Step 1. An action potential travels the length of the axon of a motor neuron to an axon terminal. Step 2. Voltage-gated calcium channels open, and calcium ions diffuse into the terminal. Step 3. Calcium entry causes synaptic vesicles to release acetylcholine via exocytosis. Step 4. Acetylcholine diffuses across the synaptic cleft and binds to acetylcholine receptors, which contain ligand-gated cation channels. Step 5. These ligand-gated cation channels open. Step 6. Sodium ions, shown here in red, enter the muscle fiber, and potassium ions, shown here in blue, exit the muscle fiber. The greater inward flux of sodium ions relative to the outward flux of potassium ions causes the membrane potential to become less negative. Step 7. Once the membrane potential reaches a threshold value, an action potential propagates along the sarcolemma. Neurotransmission to a muscle fiber ceases when acetylcholine is removed from the synaptic cleft. This removal occurs in two ways. 1. Acetylcholine diffuses away from the synapse. 2. Acetylcholine is broken down by the enzyme acetylcholinesterase to acetic acid and choline. Choline is then transported into the axon terminal for the resynthesis of acetylcholine.
Okay, so that was kind of a quick overview of those first events at the neuromuscular junction where that nerve is sending a signal down to the muscle. So how a nerve is actually able to communicate with the muscle, right? They have to do that through changes in charge and chemicals. And so we're left with this acetylcholine, right? Going out into the synaptic cleft. That's a neurotransmitter. It's one of many neurotransmitters that we have in our body, but it's the one that triggers skeletal muscle contraction. We'll see other ones when we get to the nervous system. And that acetylcholine then triggers the next events, which are going to happen on the muscle cell itself. Something else I wanted to point out about these little videos. If you're in mastering watching these videos, you will see that there are quiz questions after the video that you could use as you watch these again to kind of review the concepts and make sure that you're understanding the events. These are not graded quizzes. I don't see the results of these, but I encourage you to use them when you watch these videos on your own after lecture 2. So the next thing that we're going to look at is kind of the second step of this, which is called excitation contraction coupling. And so these are the events that happen that kind of bridge between the events that just happened at the nerve with what's actually going to happen inside the muscle cell. So we can see some of the parts here that we learned about during the anatomy of the nervous or of the muscles in the previous lecture. And so here we can see this blue area is where our calcium is stored, right? So that is our endoplasmic reticulum, and the end of it, remember specifically where it kind of gets bigger here, is that terminal cisternae. We can also see the dipping in of the sarcolemma, right? So this is our T tubule here in the center. So we're going to take a look at the events that happen as that action potential moves across the sarcolemma and down the T tubules. And so this is called excitation contraction coupling. So if you go back into mastering into the videos, you can access the second video. And this is also has a little A&P Flicks quiz that you can take after watching the video too.
Typically, a single motor neuron arising in the brain or spinal cord conducts action potentials that travel to hundreds of skeletal muscle fibers within a muscle. The sequence of events that converts action potentials in a muscle fiber to a contraction is known as excitation-contraction coupling. If we look at a single muscle fiber, we see that an action potential travels across the entire sarcolemma and is rapidly conducted into the interior of the muscle fiber by structures called transverse tubules. Transverse, or T-tubules, are regularly spaced enfoldings of the sarcolemma that branch extensively throughout the muscle fiber. At numerous junctions, the T-tubules make contact with a calcium-storing membranous network known as a sarcoplasmic reticulum, or SR. Where it abuts the T-tubule, the SR forms sac-like bulges called terminal cisternae. One portion of a T-tubule plus two adjacent terminal cisternae is known as a triad. The membranes of the T-tubule and terminal cisternae are linked by a series of proteins that control calcium release. As an action potential travels down the T-tubule, it causes a voltage-sensitive protein to change shape. This shape change opens a calcium release channel in the SR, allowing calcium ions to flood the sarcoplasm. This rapid influx of calcium triggers a contraction of the skeletal muscle fiber. Thus, calcium ions are responsible for the coupling of excitation to the contraction of skeletal muscle fibers.
Okay, so in that clip, we kind of looked at the connection between how that action potential from the nerve continues down the muscle cell, right, and is going to stimulate the last part, which is going to produce the actual contraction of the muscle cell. So the last section of this is called cross-bridge cycling. So cross-bridge cycling, when you're watching this video clip, you want to kind of pay attention to what is occurring in order for these cross bridges to form between the actin and myosin, and also what happens to end this. So how do we contract a muscle, but also how do we relax a muscle in order to stop this cross-bridge cycling? So in mastering, you're going to go back to the videos and you should see the next video, which is the cross-bridge cycling.
The contraction of a skeletal muscle generates the force necessary to move the skeleton. A contraction is triggered by a series of molecular events known as the cross-bridge cycle. In a skeletal muscle fiber, the functional unit of contraction is called the sarcomere. A sarcomere shortens when myosin heads and thick myofilaments form cross bridges with actin molecules and then myofilaments. The formation of a cross bridge is initiated when calcium ions, released from the sarcoplasmic reticulum, bind to troponin. This binding causes troponin to change shape. Tropomyosin moves away from the myosin-binding sites on actin, allowing the myosin head to bind actin and form a cross bridge. Also note that the myosin head must be activated before a cross-bridge cycle can begin. This occurs when ATP binds to the myosin head and is hydrolyzed to ADP and inorganic phosphate. The energy liberated from the hydrolysis of ATP activates the myosin head, forcing it into the cocked position. A cross-bridge cycle may be divided into 4 steps. Step 1. Cross-bridge formation. The activated myosin head binds to actin, forming a cross bridge. Inorganic phosphate is released, and the bond between myosin and actin becomes stronger. Step 2. The power stroke. ADP is released, and the activated myosin head pivots, sliding the thin myofilament toward the center of the sarcomere. Step 3. Cross-bridge detachment. When another ATP binds to the myosin head, the link between the myosin head and actin weakens, and the myosin head detaches. Step 4. Reactivation of the myosin head. ATP is hydrolyzed to ADP and inorganic phosphate. The energy released during hydrolysis reactivates the myosin head, returning it to the cocked position. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat. And as the cycle repeats, the thin myofilaments are pulled toward each other, and the sarcomere shortens. This shortening causes the whole muscle to contract. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum. Troponin returns to its original shape, allowing tropomyosin to glide over and cover the myosin-binding site on actin.
Okay. And then also that third video has a little mini quiz kind of built into it too, if you want to take that when you view the video again to see how you do on those questions. So the last diagram that I threw into your notes here is basically kind of a, I guess, a summary, a Cliff Notes version of everything that kind of happens in order for a muscle cell to contract. So these kind of 5 steps. The acetylcholine being released, the action potential going along the muscle cell, down the T-tubule, the calcium being released inside the muscle cell from that psychoplasmic reticulum, calcium binding to actin, actin's changing shape, myosin heads grabbing on, and that continues the contraction cycle. Another kind of handy graphic here that waters down what ends a muscle cell contraction. Right, so that acetylcholine has to be broken back down and removed from the synaptic cleft. The calcium has to move back into that sarcoplasmic reticulum, right? So we have to be able to transport that, calcium back into the sarcoplasmic reticulum to also end muscle cell contraction. The binding sites on actin have to get covered back up, and then the muscle cell will relax. I wanted to show you one other thing that may be helpful in your exploration of muscle cell contraction, because this is a pretty complex physiology topic here. So if you go down to the study area in mastering and you go into interactive physiology, IP, interactive physiology are some tutorials on some different kind of complex physiology topics in the course. And so if you scroll down to muscular system, you will see that there are 3 tutorials on the muscular system contraction. So there's one about the neuromuscular junction, one about excitation contraction coupling, and one about cross-bridge cycling. There's also an older version. You'll see ones below it with the same these are kind of animations that step you through, so you could also explore those. The IP2 ones are kind of the updated, newer version of these interactive physiologies, but I encourage you to explore them more, especially if this is your first time kind of being introduced to the physiology of muscle cell contraction. So it kind of steps you through what you should learn about these events. It has activities. It has a lot of like drag and drop. These are not graded. I don't see results from these, but you can use them as, and they also have little study quizzes at the end. So if you feel like you need a little more review or practice with the concepts that we just kind of went through with how muscle cells contract, I encourage you to visit those three interactive physiologies for the muscular system. And also, while we're here, there are several of them for upcoming modules too. So there's several about the nervous system, both the neuron and the synapses. There's some about the endocrine system, digestion, respiration, immune system. So some of the cardiovascular system has a lot of them. So for some of these more complex physiology topics, I encourage you to visit and check those out because they would probably be helpful to you as you're learning the physiology. Bye.